Over the past 4 years, my colleagues and I have been doing longline surveys for adult fishes out in the Gulf of Mexico to look at the toxicology that might have resulted from Deepwater Horizon for things like the incidence of skin lesions and then basically monitoring abundance. So what we've done is a survey that started down in the dry Tortugas off Florida and has gone all the way over towards Whites Bay in Louisiana. What we've done is taken the data from all of these longline sets, and these longline sets are 5 miles of main line with 500 hooks on a set. Right? We average about 50 animals or 1 per 10 hooks on the surveys, and you can see we catch a whole variety of species, including large sharks and deep water species, etc. This graphic looks at the top 40 species that we caught in our surveys, and there are a lot of, we have a lot of demersal sharks, small demersal sharks in the northern Gulf of Mexico. And then what we find is that these communities are what we call depth stratified. That means that we have a shallow water community, a mid-water community, and the deep water community. And in particular, we're worried about the deep water community because these animals like tilefish, yellow-edged grouper, and short-spine scorpionfish, those are animals that are associated with the bottom, and in the case of tilefish, they actually dig burrows into locations where oil might actually reside on the bottom. We're continuing to monitor these communities, and based on this analysis, we're trying to fine-tune the types of research we're doing so we hit the areas where we're likely to see residual impacts of things like Deepwater Horizon, and really focus on the species that are at risk as opposed to looking more generally. So we are trying to fine-tune exactly what we're monitoring over time. And this also helps us to build environmental baselines, which is so important in the Gulf of Mexico.
Well, this poster concerns how blue crabs and bay anchovies, both of which are very ecologically as well as economically important species in the Gulf, how they respond genetically to being exposed to oil. And so what we did is we experimentally exposed some juvenile blue crabs and some juvenile bay anchovies to concentrations of oil such as they might encounter after the Deepwater Horizon oil spill. And then we looked at which genes they were expressing, which genes were turned up. We are able to look at tens of thousands of genes simultaneously this way, and we see that a lot of the genes are involved in breaking down organic compounds such as found in oil. Some of these were things that were already known, but we've also discovered some new things. We'll have ways of detecting subtle changes in their physiology. Some of these things may not result in them immediately dying, but may affect them later on in life, how they grow, how they reproduce, and so forth. So with genetics, we can detect changes that are very subtle, but over the long period of time may be very important. Some of which are similar to genes that are involved in metabolizing drugs in humans. So very interesting work, very exciting to be involved in this project.
What we're looking at is during the Deepwater Horizon, the receiving station we had in Miami downlinked SAR data during the Deepwater Horizon incident. We collected data over the entire time period. And what we're looking at here in the first picture is the first SAR image that was taken 3 days after the incident. And immediately we saw that there was nearly 800 square kilometers of oil spilled from the incident. And so one of the things I'm trying to show in the poster is that this particular instrument is really good at measuring large oil spills like this synoptically so that you can see changes that are happening in 12-hour time frames. And using multiple satellites, we're able to get the complete coverage every day during the spill. And what we've done is taken that over 2 separate 12-hour time periods and shown that there's a 25% increase in area of the oil that occurred over 12 hours, and that large areas of the oil spill have translated between 15 and 25 kilometers in 12 hours. If you have a future spill, one of the things you can do is you can immediately start tracking it because we can get data within a day, we can start measuring data, and it really gives you a measure of how fast the oil is changing like no other instrument can. It really works best when you combine it with other instruments to get more information about it, but for a single instrument, it really gives you a basin-wide coverage for a large spill like this. And for smaller spills, it allows you to look at wider areas that you might not be able to do with aircraft or ships. It takes so long to get from one point to another with aircraft or ships. We can cover a very wide area, and it's very useful for telling people that we see oil here, we've seen it yesterday, this is where it's heading. Use your trajectory models to help predict where it's going to go, and send your aircraft there, send your ships there where it's going to be, not necessarily where it's been.
For this particular project, we are looking at the distribution of polycyclic aromatic hydrocarbons, PAHs, and their accumulation rate. These compounds are very important because the crude oil can have up to 10% of these compounds, and they are known for causing cancer or birth defect, or they are toxic to organisms, and they can be harmful to. So what we did was we went to this area of Gulf of Mexico and collected 17 sediment core samples, sliced them to the 0.5 to 1 centimeter interval, and looked at the PAHs concentration in each section of the sediments. At the same time, we have dated those sediment cores using lead-210 based dating so that we can know the history of accumulation of PAHs in that particular area of the Gulf. So we went back and compared their stations closest to our stations and look at their concentration, what they have found in 2010. And surprisingly, we found like even their concentration in 2010 was within the range of what we got. So we concluded the well was so patchy, probably we missed the stations, or it could be a microbial degradation or some chemical physical processes going on the sediments that has drastically reduced the concentration of PAHs on the bottom sediments. And looking at the concentration, what we can say is that this is really low, and these are they are not likely to cause any significant effect to the benthic organism living on the bottom of the Gulf.
After the Deepwater Horizon oil spill, there was a lot of flocculent material, so sea snot at the surface that rapidly sank and created a layer of sea snot essentially at the bottom of the Gulf. And what that did was actually reduce the amount of oxygen, and that has a lot of implications for anything living in the bottom of the Gulf. So anywhere from bacteria all the way up to a lot of the fish that we like to eat. So we're using this tracer to try and trace the footprint of where all that sea snot was deposited at the bottom of the Gulf. And what we're finding is that there are two zones. One of them is right around the wellhead, northwest to southeast, and the area is about 16,000 square kilometers. And then just to the east of the wellhead, about 30 nautical miles, there's another zone that's about 2,400 square kilometers. And so that gives us a really good idea of where the sea snot was deposited after the oil spill.
We study the change in the metabolome of the deep sea coral Leiopathes glaberrima in response to exposure to oil and dispersant. And since this coral also has two color morphotypes, red and white, we also saw if there was a difference in the metabolome between red and white coral. So this first figure is just a description, an illustration of the difference in the color morphotypes. So you can see the red coral is pretty red, it's like blood red, and then the white coral is pretty pale. The next figure shows that we were successful in extracting and separating and quantifying our metabolites. So we get a sense of the metabolome of our coral. And here I have a few examples of the metabolites that were identified as different between our treatment groups. So this particular metabolite that was identified as C17 sphinganine was identified to be lower in abundance in the treatments with dispersant. So that we had 3 treatments: the treatment with oil alone, dispersant alone, and a treatment with oil and dispersant. So in the treatments with dispersant and oil and dispersant, we found that this metabolite, C17 sphinganine, was lower in abundance. And C17 sphinganine is involved in lots of biological functions such as apoptosis and cell signaling. So the dispersant might be having an effect on these cell functions. It's an interesting story that's starting to emerge that the dispersant seems to might have an effect on the coral's metabolome.
We're looking at the trophic structure and the feeding ecology of hackfishes from the Gulf of Mexico and also at their mercury bioaccumulation. And we're trying to piece apart which feeding role they have in the deep sea food web, whether they're scavengers or actually predators and hunters. And we're also looking at mercury concentrations of the fish and see if there's any variation from the oil spill out or after the oil spill. Well, hackfish are actually really interesting. They're very poorly studied in the Gulf of Mexico. We don't know a lot about them aside from descriptive stuff. We don't know anything about their life history or ecology, and this was the first step for me into getting into that field. Because they're associated with the seafloor, they burrow into the sediment, they could potentially be heavily affected by the oil spill if there was any real increase in methylation of mercury from the substrate. And we're also seeing increase in trophic level with depth, but perhaps the most, as well as mercury increase with depth and trophic level. Perhaps the most interesting thing is that their inorganic mercury concentrations are not like the usual 5%. They actually have a lot more higher inorganic content, and we don't know what that is, so that's something that we would like to look into. In this project, I'm looking at the microbial ecology associated with sand patties that were collected on Fort Morgan and Gulf Shores beaches as a result of the Deepwater Horizon spill. So I've been looking at the 16S community of these sand patty samples as well as the metagenomic potential, and additionally looking at if they're possibly able to be further biodegraded once they're deposited on the coastal beaches that we collect them on. In this project, I've just looked at single tar balls from these 2 locations, and I'd like to look at maybe replicate tar balls in the future to see maybe if the community in a tar ball or a sand patty is the same at one location, or maybe every individual tar ball has a different microbial ecology associated with it. When we were first introduced to these sand patties and what they were, we had Christoph Eppley from Bigelow show us what these samples were like, and these are the locations he had traditionally sampled. So we kind of wanted to continue sampling in these locations to maybe get an idea if there's some consistency. The ultimate location and fate of these tar balls is ending up on the coastal beach environments. I think it's important to understand what's happening to them and if they're able to be biodegraded by the bacteria that's associated with them, and what impact they have on the beaches in terms of the microbial community that can degrade these hydrocarbons.
So this poster is really looking at the concept of transport timescales, how long it takes, what the timescales are for something to move from one place to another. And this idea is in terms of the age, which is the time since it was last at a source region, and the residence time, which is how long it stays in a given region that we're interested in. So we're looking at this in the context of not only the Gulf of Mexico, but the whole Western Atlantic Ocean as well. We have kind of 2, as I mentioned, we have the age and the residence time, which are these 2 concepts that we can relate to transport timescales. And the age is what we see in the top panels here, which basically tells us how long it's been since water was last at river sources. And we isolate this not only into rivers from all over these Americas, but we also isolated just the rivers, just to the Mississippi and Atchafalaya River. And we find that the Mississippi Atchafalaya River can actually reach places like like North Carolina in less than a year. In terms of the residence time, in the Gulf of Mexico, we're looking at how long water stays on the continental shelf, and we find that really the most variability is right on the Texas-Louisiana shelf, where we find that in the summertime, water stays there a lot longer, on the order of 9 to 12 months, whereas the rest of the year, the water stays on the Texas-Louisiana shelf for maybe only 4 to 6 months. If we can understand these timescales of how long water stays in these given regions, for this example, how long it stays on the shelf, we can help biogeochemists understand what some of the background timescales are for the biogeochemical processes. And we can understand how long water, say oil that got on the shelf, how long it'll take that water to leave the continental shelf and get back into the deep ocean.